Hello, and welcome to 11.2. In this lesson, we're going to talk about pharmacodynamics. So the last time we talked about the different kinetics, and really the key points to take home from that lesson were ADME, absorption, distribution, metabolism, elimination. And those are the four parameters that we take into account when we're trying to determine how well a drug is going to absorb and be distributed and thus reach its target tissues. But now we're going to shift and we're going to talk about how we evaluate and determine how well a drug actually is going to work. And by how well it's going to work, we mean how is it going to achieve its efficacy. And when we talk about efficacy, really we're talking about the amount of biological response that it's going to produce. And there's other things we need to think about as well, including safety determinations and some things we'll talk about later. Okay, so the mechanism of action of the drug, that is really what pharmacodynamics is all about. The mechanism of action, how does it achieve its effect? And how well does it achieve its effect? So not only what is the mechanism of it, but how much of an effect does it produce, okay? Now, there's actually many different ways that drugs can affect neurotransmitter systems, that they can affect cellular excitability. Remember, when we're talking about the central nervous system, we're talking about neurons, and we're talking about being able to modulate or affect the function of neurons. So increase the rate of firing, increase the amount of neurotransmitter that's available at the synapse, or decrease the amount. Okay, so these different ways that we can do this, we can affect the synthesis of neurotransmitters, we can affect their storage. So if they're made but they're not stored in vesicles, then we're not going to be able to have vesicle release into the synapse. So that's one way. The actual release, so the trafficking of the vesicle to the membrane and the release, the reuptake of the neurotransmitter. So if we block it from being reuptaken, it's going to be present in the synapse longer. And catabolism. And by that, really, we're talking about metabolism, but specifically, we're talking about breaking down. Okay, breaking down the neurotransmitters. So the important thing to remember here in this section is that, and really through all of pharmacology, anytime you're thinking about pharmacology, is that no matter how selective we think a drug is on a particular type of receptor or on a particular process, drugs are not completely clean, okay? And in fact, that is the reason why anytime you are prescribed a drug or you pick a drug up from the pharmacy, there are always side effects, because depending on the concentrations of a drug, the more drug that you give, the more and more it will start to bind to different receptors or different signaling molecules that it's not intended to bind to. Okay, it's concentration specific. And even at the recommended dosages or concentrations, right, there's still going to be some non-specific effects, because drugs, again, they are in the circulation and they are distributed all over the body. So you're never going to be able to limit a drug to one very specific place in the body. They're going to bind to different places and produce effects. So we're going to start out by reviewing briefly these different points of attack, if you will, different targets of drugs, but we're really going to focus on a couple of key terms, and that is, what do we mean when we're talking about an agonist versus an antagonist? What is an agonist and what is an antagonist? So at this point, I'm going to show you an animation, and we're going to use an ionotropic receptor as our sort of model receptor, and we're going to talk about agonists and antagonists and some different types. Okay, so to help us understand agonists and antagonists, I thought I would incorporate a animation, because animations are always good and they help us out a lot. So here you can see a synapse, and this is showing us essentially all the steps in synaptic transmission. Now, drugs can act in many different points along the pathway to either increase cell excitability or to decrease it. Now, we know that there's a whole myriad of different components here. So we have to have synthesis of a neurotransmitter first, then we have to have storage of the neurotransmitter from some activation of the cell has to then move to the membrane and fuse and release into the synaptic cleft, and bind to receptor of the postsynaptic neuron. So there's different points along the way that we can affect this. But in general, when we talk about an agonist and an antagonist, what we mean is essentially something's ability to mimic what the endogenous ligand would do in this place. So the endogenous ligand is the ligand inside the normal body that actually is supposed to bind to the receptor. So for example, for the nicotinic acetylcholine receptor, the endogenous ligand is acetylcholine. Now we know that nicotine will also bind to this receptor, right? But nicotine is an exogenous ligand. It is a ligand that will bind to the receptor, but it comes from outside of the body. It's not something that we normally make, okay? It wasn't nature's plan for our bodies to bind nicotine to this receptor, okay? So these are the sort of words that we're going to use. Now, any drug that enhances or mimics the effect of the endogenous ligand is what we are going to call an agonist, okay? So that is a drug that mimics the endogenous ligand, okay? And likewise, an antagonist is going to be any drug that blocks the effects of the endogenous ligand. So whatever the endogenous ligand would normally do in this situation, the antagonist is going to block that from happening. Okay, so agonist mimics the normal effects, the antagonist blocks the normal effects. Okay, so we're going to use the example of a normal ionotropic receptor. So remember, this is a receptor that has the sort of barrel structure, tetrameric or pentameric, that goes through the membrane. So it's the transmembrane barrel that has an ion channel that will open or close in response to activation. So here's our channel, and we're going to pretend we can just call this GABA, okay? So in a normal situation, the endogenous ligand is GABA, and the endogenous ligand would bind to this receptor, right? And in a normal situation, this would cause the channel to open, and it would cause ions to flow into the receptor. Now, in the case of the GABA A ionotropic receptor, this would mean that chloride ions, all right, so Cl minus, are going to flow into the cell, and it would induce a hyperpolarization. So it would limit the excitability of that cell. It would decrease the chances that it's going to fire, right? This is just our example. It's helpful for us to have one. Okay, so in the event that we actually have an agonist, so we have a drug that's acting as an agonist in this situation, what the agonist is going to do is it is going to bind, and it's going to produce the same result as the endogenous ligand would produce. So in this case, we have this exogenous ligand that just bound to this site on the receptor and caused the channel to open, just like GABA would. Okay, and it's going to cause this to happen. Okay, because it comes from outside the body. Again, we call it an exogenous ligand, and that's really what we're talking about when we're talking about drugs. Okay, they're exogenous ligands. They're going to bind to different places on a receptor. Okay. Now this in particular, and what we're going to talk about, is the difference between an agonist and antagonist in something called competitive or non-competitive. Okay, so if you can see here, this ligand actually bound to the same site that the endogenous ligand would normally bind to. Okay, now we're going to look at the next situation. Here we have a drug. Okay, this is an antagonist. Again, it's an exogenous ligand. It binds to the same site that the endogenous ligand would bind to. And in this situation, we can see that what it does is it actually prevents the endogenous ligand from binding. So even though this binds to the same site, it binds in a way that produces the opposite effect of what the exogenous ligand would produce, because now the channel cannot open. This drug, this antagonist, has blocked the opening of this channel because it is bound to the same site that the endogenous ligand would bind to. And therefore, it's blocking the way, okay? Just picture it like you're trying to get through a doorway and somebody's standing in front of you. They're blocking the entrance to the doorway. That's essentially what this does. And this is what we call competitive antagonism. So this is a competitive antagonist because it's competing. It's competing for the same binding site. It binds at the same site, okay? Now, we're going to move into a different type of situation. So now we have a non-competitive ligand. The reason why this is non-competitive is because it's actually binding to a different site, okay, than the endogenous ligand. So this ligand is not competing with GABA, because GABA would come and bind right over here, right? And this ligand is binding over here. So it's binding to a different site. It is non-competitive, okay? And we can have different effects from a non-competitive ligand, okay, or a drug. So we're going to look at this scenario a couple of different times. So here we have what we call a non-competitive agonist. So this is an agonist, so remember it's going to produce the same effect as the endogenous ligand and activate the channel. We can also have a non-competitive antagonist. So this one is going to produce the opposite effect of the endogenous ligand, and it's going to block the channel from opening. So you saw we had two different scenarios just now. We had a non-competitive agonist. So again, the key for any agonist is that it's going to perform the same function that the endogenous ligand performs. So in this case, it's going to do the same thing that GABA does when it binds to this receptor. Okay? The antagonist is going to do the opposite. It's going to keep it from opening. Now we can see here that this is a non-competitive antagonist because it binds to a different site from where the endogenous ligand binds to. Okay? All right. So hopefully that was good, and you were able to see through the animation how we can have these different types of ligands. We can have these exogenous ligands. That's really what drugs are. They're exogenous ligands that are introduced into the system and can bind to receptors, and they can bind as an agonist, so mimicking the effects, or as an antagonist, which is blocking the effects. And there's lots of different ways that we can affect neuronal excitability, i.e. whether or not a neuron is likely to release neurotransmitter and whether it's likely to activate the next cell by releasing neurotransmitter. So I went over a couple of these in the video, but just to kind of show here, we can affect the level of synthesis. So remember, we have biosynthetic enzymes and we have neurotransmitter biosynthesis that's regulated by these enzymes to produce neurotransmitter. So if we can either increase the synthesis or decrease the synthesis, we can affect the excitability. So we can do this with agonists or antagonists. We can also interact at the level of storage. So remember, the neurotransmitters, once they're made, they're stored in vesicles. These vesicles then need to move to the membrane. Some stores of these vesicles are already near the membrane, and some of them are further away, and they actually need to traffic towards the membrane in response to an action potential in the presynaptic neuron. And so we can affect storage, or we can affect the release to different points to either increase the rate of neurotransmitter release or to block neurotransmitter release, whether we're talking about an agonistic or an antagonistic effect. All right. And another popular way, we're going to talk about this a little bit when we talk about depression and serotonin, is blocking the reuptake. We're also going to talk about this when we talk about some drugs of abuse as well. But blocking the reuptake of neurotransmitter. And like I mentioned, the neurotransmitter gets released into the synapse, right? Well, if we block the reuptake, that means that this neurotransmitter, assuming that it's not all going to get degraded in the synaptic cleft, because remember, some neurotransmitter does in fact get degraded by degradative enzymes such as monoamine oxidase or acetylcholinesterase, but that despite the fact that some of it's being degraded, we're still going to have a bunch of it that would normally get reuptaken, right? That's not going to be reuptaken and is not going to be degraded. It's still going to be present in the synaptic space and able to bind more to the receptors, okay? Or speeding that up, it would do the opposite effect, okay? And finally, receptor binding directly. So we can have an agonist that binds directly to a postsynaptic or a presynaptic receptor. We didn't talk much about presynaptic receptors, but we've got those too, okay? These are often called autoreceptors, and what they do is they tell the cell something about how much has been released, and they sort of, in most cases, serve as negative feedback, okay, to maybe block more synthesis of the neurotransmitter, because we know now that it's present in the cleft. And so even though a bunch of it's really going to bind to the postsynaptic cell, some of it might bind to the presynaptic cell as well on these autoreceptors and tell this cell, okay, we've already dumped a bunch of dopamine out into the space. We don't need any more dopamine release, right? And so maybe the cell starts to downregulate the synthesis of dopamine, or starts to downregulate the packaging of dopamine into vesicles. And all of these things are complicated, and they work in concert with one another to regulate the amount of neurotransmitters that are being synthesized and released from the cells. Okay, so the mechanisms of action, just to go over there again, we have the agonist, okay, mimics the effect of the endogenous ligand. The antagonist blocks the effect of the endogenous ligand, right? So here we can have, we see that we have a receptor site. Now this is what they call natural chemical here. This is the endogenous ligand. It's binding to a receptor. So an agonist here we have that's also binding to the same type of receptor. So here this is essentially like there's several receptors of the same type on the membrane here, right? So one of these is bound endogenously, the other one's bound by our agonist, and it's going to increase cellular activity. Whereas here we have the antagonist, it's here going to fill up this space and it's going to block the cellular activity, all right? Now, competitive versus non-competitive, again, hopefully this was illustrated well in the animation, but just to review it, okay, the competitive means that we're binding at the same site as the endogenous ligand. And if something is non-competitive, it is not competing for that site. Okay? Remember, competitive means that we're competing. We're competing for the site. So they both want the site. Okay? The drug is going to be trying to get to the same site that the endogenous ligand gets. It's competing for it. Non-competitive means it doesn't care. All right? So non-competitive means that it's binding to a different site. And we defined in previous lesson that this is called an allosteric binding site. Okay, it's an allosteric site because it's different from the one that the ligand normally binds to. Okay, so here you can see an antagonist here. This is a competitive antagonist because it's filling the space that the endogenous ligand would normally bind to. And here we have a non-competitive antagonist. So it's blocking the opening of this channel, right? Again, this would be like something we use the GABA example, so let's just stick with that one. If this was GABA and this is supposed to induce opening of the channel, this non-competitive antagonist is blocking the opening of that channel, but it's doing it because it's bound to a different site. Okay, and this is an allosteric binding site. Okay, now we can have different types of agonists. So the situation actually gets a little bit more complex. We can have something called a partial agonist. What this means is that it's going to bind to the receptor, okay, and it's going to produce an effect, but that effect is not going to be as robust as the endogenous ligand's effect would normally be. So let's say that the endogenous ligand would cause the cell to fire several times. Maybe the partial agonist binds it and only causes the cell to fire one time. Okay, and we're going to see this more when we look at the curves. So for now, just understand that the partial agonist, it produces the same response as the endogenous ligand, but the magnitude of the response is lower. So it doesn't produce the maximal response, it only produces part of the response. Okay, and an inverse agonist is a little bit trickier, and this elicits the opposite response. I don't want you to think too much about this one because this is a little funny, and you have to know a little bit more about pharmacology to understand exactly how these work. Okay, so inverse agonist, it's good to know that it's out there. Okay, it does bind to the same receptor and it produces the opposite effect, but there's other reasons for that that we're not going to get into. Okay, now here we can measure the magnitude of an effect in a biological system by something called the dose response. Okay, so we talked a little bit about this before. The dose response allows us to measure the magnitude of an effect. So here you can see that there's three curves. Okay, we're using a logarithmic scale here, and this is actually, this curve was generated in response to an opioid drug. Okay, so on the y-axis, what we're measuring is the percent of receptor activation. Okay, and there's ways that we can do this. So if we for example, we can grow a bunch of cells in a dish, and we have ways of knowing exactly when all of the receptors are activated, or half of the receptors are activated, etc. And we can measure this in response to different concentrations of a drug. So maybe we have a whole bunch of different conditions, and in one of them we add, I don't know, 100 nanomolar concentration. The next one we have 10 micromolar, then we have 500 micromolar, then we have 1 millimolar, right? So we can increase the concentrations and measure the amount of receptors that are activated, okay? And this generates a curve for us. The point here is that if we look at receptor activation, we can see that a full agonist, so this full agonist just means it's a normal agonist. It's capable of producing the full magnitude of response, okay? So if we give enough, if we give a high enough concentration, we will eventually activate all of the receptors, okay? Remember, it's concentration dependent, so we might have a bunch of receptors present in the system. If we only add a little bit of drug, we might not have added enough to bind to all of the receptors. So our effect might be less than maximal. But if we continue to add concentration, okay, eventually we will bind all of the receptors and we'll achieve our full effect, okay? Now here, we can see that we have a partial agonist, okay? And the reason why this is a partial agonist is because even at the same concentrations, okay, we only achieve about 35% of the receptor activation here, even though we keep on adding lots and lots and lots of this, okay? And eventually it levels out to the point where we continue to add higher concentrations, but we do not get any further increase in the amount of receptor activation. So this is showing us that we have an agonist here that it does achieve an effect, because we can see it here, but it's not as high is the actual agonist itself. So this is a partial agonist. And this is in direct contrast to the antagonist. So here we can see an antagonist curve, and it's exactly right on top of the x-axis, okay? There is absolutely no effect. So in the presence of this antagonist, okay, we have absolutely no receptor activation. So this is showing us that it is in fact blocking, okay? It's blocking the response. Now, more on dose-response curves. We can measure these things, and we can see how antagonists and the different types of antagonists are actually affecting things in our system, okay? So here again, we have percent of maximal response on the y-axis, and we have the dose. And again, we're working with the logarithmic scale here on the x-axis. Now, you can plot this in a couple of different ways. A lot of times we use a semi-log scale, like the one shown here, because it's easier for us to achieve this sigmoidal curve, and it tells us something about our effective concentrations, but we're going to talk about that later. For now, just know that this is the most common method of actually showing a dose response in a drug experiment. Okay, so here we have an agonist and it's producing the maximal response. And we can see that it's producing the maximal response because what happens? Well, we have a steady increase until it plateaus here at the end, right? So this is 100% maximal and it's going to reach the plateau. This is the agonist by itself, okay? Now we have another situation where we have the agonist present, but instead we have put a competitive antagonist in the mix, okay? So let's say we have a bunch of cells that we've grown in a dish, right? And we've given an agonist that's going to activate these receptors, and it's going to produce a response. And we can keep increasing concentrations of the agonist until all of the responses is met. So we've achieved 100% of our response. Now we can take different wells that contain cells, the same types of cells. We're going to add the same type of agonist at the same concentrations, but we're also going to include an antagonist. Okay, and in this case, a competitive antagonist. So the competitive antagonist, remember, is going to compete for the same binding site as the agonist. So what does that mean? Well, that means that if the competitive antagonist binds to the endogenous ligand's binding site, okay, the binding site of the agonist, what's going to happen is it's going to block the agonist's ability to bind it. It gets in the way. Remember, competitives get in the way. So the only way to stop this from happening, okay, and to beat this competitive antagonist at their own game is to ramp up the concentration of our agonist, okay? And so what we can see here is we can tell that we're dealing with a competitive antagonist because our curve for the percent of activation is going to shift to the right. Well, what does that mean? What that means is that if we continue to add more and more concentration of our agonist, eventually we will get to a high enough concentration where we will start to beat the competitive antagonists at getting to the binding site. Okay, so we'll be able to knock this guy off of the binding site because we have so much agonist present okay, that now we can get there first and we can get there more often, and the competitive antagonist no longer can block the response. So notice we still achieve the same maximal response with our agonist, but what is the key difference? The key difference is that if we have a competitive antagonist present, we need more. Okay, so this is the dose. This is the amount of the drug that we have to put into the system. And here we can see that before, where our maximal response was achieved here at about 10 on the log scale, now we're up to about somewhere between 100 and 1,000, okay, to get the maximal response. So we can still get the maximal response, but we just have to give a lot more drug, all right? Now, let's look at a different scenario. This is what happens when we add a non-competitive antagonist. Now remember, the non-competitive antagonist, it's not competing with the agonist for the site, because it's actually going to bind to an allosteric site. It's binding to a different site. Okay? So what that means is that in the case of our agonist alone, we're going to produce a response. Now, if we add a non-competitive antagonist, we're still going to get some kind of response, okay? But what's going to happen is we're going to have a decrease in the magnitude of the response, okay? We can't reach the maximal response because no matter how much agonist we continue to add, what's going to happen? Okay, we're still binding those endogenous sites, but the problem is that the non-competitive antagonist is changing the receptor function by decreasing the activation on a allosteric site. So it's binding at a different site. So no matter how much agonist we add, it's never going to knock this off because this is bound in a different place. Okay, different place on the receptor. So this is non-competitive antagonist is actually going to decrease the response, okay, in our system. So two different things that happen. This is cool to know about, and it's relevant because this is a lot of times how we can determine what types of drugs we're going to use, an agonist versus antagonist, and what type of antagonist we want to use, right? So competitive antagonist, you're going to see a shift in the right, shift to the right of the dose-response curve. So it shifts right, same magnitude of response, but it requires much more agonist to knock this off of the binding site, right? Whereas with our non-competitive, we're decreasing the magnitude of the response, okay? Now, how do these things tie in with efficacy and toxicity? What do these things mean? Ultimately, with a drug, what we want is something that works really well and is very safe. It doesn't produce a lot of toxicity. And I like this cartoon here, okay? And he's saying, yeah, some hideous monster was not one of the adverse reactions that we were expecting. Okay, this is funny because, in a strange sort of way, we have a whole system that we go through. Okay, there's a whole sequence of events, a long, arduous sequence of events from where we start with a candidate drug that we think might be able to help with something. And we start with it in a lab. And usually we start with it in a dish or in an animal model. Okay, so from that point to the point where we actually have a drug that we're giving to human patients, okay, in a controlled, blinded clinical trial. We'll talk about that in a minute, okay? But basically testing it in humans to see whether or not it's still going to be effective and safe. This process can go on from, it can take 15, 20, 25 years sometime from beginning to the end by the time that a drug is actually approved for human use and costs billions of dollars, okay? So the question becomes, how do you know? How do you know that a drug works, and how do you know that it works well? What are the criteria that we use to determine this? Well, we start with preclinical testing, okay? Preclinical testing means that pre, meaning before, and clinical is, in the clinic, so that's human patients in some sort of hospital type environment, right? So preclinical means it's before that, okay? And in preclinical testing, what we're using is we're using what we call in vitro, which essentially means in a dish. So this is where we grow up cells and we measure the effects of drugs on particular parts of cellular function, okay? So for example, on receptor activation, the ability to upregulate, okay, or increase the production of a certain protein, or maybe the ability to increase protein kinase activity, okay? If we give this drug in neurons in a dish, does it actually increase phosphorylation? of certain types of proteins. So these are all different types of things that we can test, and there's millions of things we could test, right? You can ask questions all day long. This is the in vitro, okay? We also have the in vivo studies. These are still preclinical because in vivo, vivo meaning life, right? So even though they're in living organisms, they're not in people. Okay, so in vivo preclinical studies typically mean that we're using lower level species. And usually we start off with things like, well, I guess it sort of depends. A lot of the earlier experiments that were done with uncovering the function of neurons in terms of action potentials and their electrical conduction and such were done in the squid. Typically, when we're evaluating higher level sorts of outcomes, okay, so if we want a drug that's going to help cure some sort of disease, right, whether it be drug addiction or it be Alzheimer's disease, some kind of neurodegenerative disease, or even injury to the brain, okay, we're going to be actually using species like rodents. So typically we start with mice or rats. You then might move up into higher level species like a sheep or a pig, okay, and you do some testing in those species, and then eventually, once all the preclinical testing is done, and you see that you have a compound that works and it seems like it's pretty safe, then you can move to clinical trials. And in the clinical trial, that's when we're actually going to do the final testing, okay, to make sure that it's actually safe and efficacious. Efficacious, right? The efficacy means that it works. So efficacy is really the ultimate measure, and that is it actually producing the biological effect that we wanted? Okay, i.e., does it reduce tumor growth? Okay, does it stop? Does it improve outcomes in Alzheimer's patients such that the progression of the disease is slowed? Okay, does it improve vascular function? So does it improve blood flow? Okay, after a stroke. These are all efficacy. This is, does it actually work? Okay, but there's really, throughout the course of getting to this point, there's some really important factors that are evaluated, okay, and this is basically to figure out whether something is safe and how well and how much the biological effect is actually working. So are we producing the effects that we want? Okay, so we already sort of talked about this a little bit in terms of the dose response, but I want to add a couple of terms. So here we're looking at the percentage of the biological effect, and remember the biological effect can be many different things. We could be talking about the amount of phosphorylation, okay, the amount of kinase activity. We could be talking about receptor activation, or we could be talking about something like the amount of the slowing of tumor progression, something like that, right? So biological effects could be many different things, okay? But we know that we have an effect, and we have something here on this line that's called an EC50, okay? So the EC50 is what we call the effective concentration, okay? So this is the concentration at which we have achieved 50% of the maximal effect, okay? So the maximal biological effect, when we've gotten 50% of that effect, the concentration where that happens is what we call the EC50. Okay, so this is a very standard measure of efficacy, of the biological effect of something. Okay, it's called the EC50, effective concentration. Okay, so this EC50 tells us something about efficacy. Another thing that we really want to hammer home in these early experiments is the selectivity. So selectivity of a drug means how well it is at actually binding only to the target that we're trying to bind to. Okay? For example, we talked a little bit about ethanol in class. Ethanol is a good one because it's a small molecule and it binds to lots and lots of different stuff. Okay? So ethanol is not an extremely selective drug. Right? Ethanol is going to go in, that's why it produces so many different effects on the system. It's going to bind in many different types of receptors in many different areas of the body. Now, we might want a drug that only binds to a very specific type of receptor on a very specific type of cell in the body. Okay? Now, selectivity means how well it does that. Does it actually only bind to this particular type of receptor? And selectivity really will break down for most drugs at some point, and that point is when you give too much of it. Okay, so the more you increase the concentration of a drug, no matter what drug it is, it's going to start binding non-selectively to other things. Okay, and a lot of times this is what we refer to as side effects. Okay, it's non-selective binding to other types of things that produce different effects other than the one we're trying to achieve. Okay, so selectivity is important. Potency is also very important. So the potency refers to how powerful our effect is, like how powerful our drug is in producing this effect. Can we produce the effect by using as little as possible? Okay, that's the way you can think about it. And in fact, this is directly tied to the affinity. Okay, so the affinity of a drug tells us how well it actually binds the receptor. Okay, and I use the word tenacity here. I like that word because drugs will bind on and off to a receptor. Some will stay bound for longer, some will stay bound for shorter, some will come off and on, right? So affinity is how strong of an attraction it actually has for the receptor, okay? So something that has a very high affinity for the receptor is going to bind a lot better, okay? And it's going to improve the overall effects of our drug, okay? So we talked about the EC50, all right? We also have IC50. Now, the only difference between this is the inhibitory concentration, okay? So the IC50 is what's used if we're dealing with an inhibitor. Okay, so if we're giving something that we actually want to block a biological effect, okay, so that's the same thing, okay, it's just the effective concentration or the inhibitory concentration. So the concentration that inhibits 50% of the maximal biological effect, okay. We also have what we call the ED50 and the ID50, okay. Now the only difference between the EC and IC and the ED and ID is whether or not we're dealing in vitro or in vivo, okay? Whether we're dealing in a dish with cells or whether we're dealing in a live organism. So the C refers to concentration, and when we're talking about how much of something we give, if we're giving it to an experimental system that's not alive, i.e. a bunch of cells in a plate, okay, we talk about the amount in terms of concentrations, all right? If we're dealing with an actual living organism, we talk about the amount we're giving in terms of dose, okay? So when you see ED50, that's the effective dose, okay? Or the inhibitory dose, all right? Same thing. It's just dose if it's in a living organism or concentration if it's not in a living organism, all right? So that's the idea there. But what we can see if we examine this a little bit closer is we have 4 drugs here, A, B, C, D, okay? Now we have the percent of the response. Again, we're looking at the biological effect here, and we have a semi-log scale for concentration on the x-axis here, okay? And what we can see here is that the EC50 for these 4 drugs is not the same, okay? Now they both produce the same maximal effect, right? So they both every single one of these drugs, all 4 of them, eventually gets to 100% maximal efficacy, right? But the difference here is how much we need to get there, okay? So here, the EC50, the effective concentration, okay, for drug D, so the amount, the concentration we need to get 50% of the maximal response is way higher in D than it is in A. So you see here how A has a much lower EC50, okay? The concentration required here is much lower for A than it is for D in receiving half of the maximal effect. So this is to say that A is much more potent than D, right? It has higher potency because we need less of it to achieve the same effect, okay? The reason why we need less of it to achieve the same effect is because it has much higher affinity for the receptor. Okay, so it has a much higher, a better binding profile. It's going to bind to the receptor much more, or maybe for much longer. Okay, so it has a higher affinity and it has a higher potency. All right, does that make sense? So, in contrast, we can take a look at another example. Now we have 3 drugs here, A, B, and C. Now each one of these drugs has the same EC50. Okay, so we can see that the EC50, the concentration that's required to achieve, okay, 50% of the maximal response of this drug is the same for each one of them. But what is the key difference here? Tick tock. Tick tock, tick tock, tick tock. Okay, I'll answer it for you. Efficacy, all right? So the maximal response for these 3 drugs is not the same. And actually, sorry, these 4 drugs, because I see we got D way down here at the bottom, okay? It's not the same. A produces a much larger maximal effect than does B, which in turn produces more than C, and C producing more than D, okay? So even though the EC50s are the same for these drugs, okay, because the same concentration for all of them achieves half of their maximal effect, the maximal effect is different, right? C doesn't work as well as A. It doesn't produce as much of a biological effect as A does, okay? Now, in terms of our different types of agonists, what do you think this tells us about the types of agonists we're looking at here? If you were tending towards full agonist or complete agonist versus partial agonist, you are exactly, perfectly on the right track, okay? Because B, C, and D are definitely partial agonists because they produce a response, but they do not produce the same magnitude of response, okay? A produces a full response, B, C, and D produce only a partial response, right? Now, the last thing that we need to consider is safety. Now, again, we're going to have these 50s, all right? So TD50 and LD50 are what we use to measure safety, okay? Now, the T for the safety measure stands for toxicity, okay? So this is toxicity dose, right, or toxic dose, 50, and LD is lethal dose, all right? So you can imagine, right, we typically don't measure LD50s, the lethal dose, in human studies, right? Because if a person died or people started to die from giving a drug, the drug would immediately be discontinued for ethical reasons, right? But in animal studies, we do measure lethal dose, okay? Because oftentimes in a lower level species study, the study will be continued if it's not clear exactly what's happening, and so you will get these LD numbers sometimes there, okay? You won't get them in a human study. But in humans, we do have toxicity, right? So you know that when you take a drug, there are side effects associated with this drug, right? Those side effects really are toxicity, okay? And we always make a determination as to how much toxicity we're willing to put up with, okay? How many side effects we're willing to put up with, and how severe, okay, we're going to permit them to be. Now, you can think of it this way, all right? If you have a terminal illness and the prognosis is negative and it's not expected that you're going to be able to survive much longer, you may be willing to take a drug that has more toxicity, okay, if it means that you actually have a chance of improving, or extending the amount of time that you're going to be able to live your life. So there's always a trade-off between these things, right? For relatively minor sort of ailments, we're probably not willing to put up with a lot of toxicity, right? You probably wouldn't want to take a medication for a headache if one of the side effects was that it was going to cause severe bleeding, right? It's probably not worth the trade-off there. Okay, that toxicity would be much more of a negative effect than actually dealing with the headache itself, right? So these are always considerations that come into effect. But here we can look at in the same way, okay? What we come up with here is what we call the therapeutic index, and this is what's used to determine the safety of a drug. Now, the therapeutic index really is just a ratio, okay, between the safety and the efficacy of a drug, okay? So the efficacy, remember, here's the ED50. So this is sometimes, we'll call this a therapeutic effect, right? So therapy, we're actually helping something, some kind of disease or ailment. So the therapeutic effect, this is the biological effect, okay? So ED50, remember, is the effective dose, the dose where we've achieved 50% of our maximal biological effect, right? So this is good. The lower the ED50, the more potency you have, right? And then we're looking at our TD50, and this is the toxic dose 50, okay? So the dose at which we've achieved 50% of the maximal toxicity, okay? So what we want here is a pretty big space, all right? The larger the difference, the better off you are, all right? If you have a very narrow window, okay, between the safety and the toxicity, okay, that's not usually a good thing, because that means that you have very little wiggle room in between the dose that you need to achieve a beneficial effect and the dose that's actually going to start causing toxicity, right? So that's normally not what you want. This ratio from safety to efficacy, okay, we want this to be low. So this is essentially the whole take-home message between, or rather, behind how we get to determine whether or not something should be used. So we have a drug, we've been trying to develop it, we're trying to use it to treat schizophrenia. Is this a good drug or a bad drug? Well, how do we know? These are the reasons why we know. Is it selective? Is it potent? Okay. Does it produce a robust biological effect that we want? Does it limit the amount of toxicity? Okay. And the way that we can tell this is by looking at all these different parameters to do that. All right. So good luck on your quiz, and I hope you enjoyed this installment of neuropharmacology. And next we will be talking about some different types of drugs, and we'll be getting into some different types of injuries and disorders. All right. See you next time.